Hi, thanks for introducing that talk. My name is Ksenia. Currently, I'm a postdoc in Aston University in Rotterdam. But today, I'm going to present my PhD work that I've done at the University of Birmingham in Brain Lab. And on the slide, you can see an organism I was working on, E. coli. On the bottom of this bacteria, you can see several flagellas. A flagellum is a motility organelle that allows the cell to swim through the liquid towards food or away from toxic chemicals. And in my project, we found a new mechanism of transcriptional activation of promoters that regulate several important ingredients essential for flagellar biosynthesis. So the talk is going to be in 4 parts. I'll give you a quick introduction to transcription initiation in bacteria and introduce you to certain factors. Both of them relate flagellum, and I'm going to explain how. Also, I'm going to introduce you to a transcriptional factor that activates antibiotic resistance in bacteria and try to connect antibiotic resistance with flagellar biosynthesis. And towards the end, I will show you a mechanism of transcription activation. So let's briefly remind ourselves how gene expression works. RNA polymerase, or RNA for short, activates genes via recognition of the very conservative promoter elements, -10 and -25. Once the promoter has been recognized, gene can be expressed, and RNAP is made. Sigma factor plays a major role in transcription initiation because, first of all, it sends signal like RNAPs to different parts of the chromosome to transcribe different sets of genes required under different circumstances. Secondly, sigma factor melts promoter DNA upon binding, and this melting is very important for transcription activation. Please remember that, as I will come back to it towards the end of my talk. So E. coli has 7 different sigma factors. However, only 2 of them are going to be important for us today. Housekeeping sigma in green that directs transcription of early flagellar genes, and flagellar sigma that directs transcription of late flagellar genes. And this is a schematic of flagellum. It consists of basal body, hook, and filament. Transcription of all of those components regulated in a cascadal way by those 2 sigma factors. So basal body and hook are transcribed first by housekeeping RNAP and FLGC transcriptional activators. Both of them activate flagellar sigma that direct RNAP to the genes required for the completion of flagellum. And now it looks like we have a nice clear separation between those 2 sets of genes. However, in my experiments, we found that transcription of the upper part of the basal body and hook can be regulated by those 2 sigma factors. And now I'm going to tell you a little bit more about the genes that encode these structures in flagellum and their promoters. So the upper part of the basal body and the hook are encoded in FLGB promoter. FLGA encodes chaperones and flagellar anti-sigma factor. And before we started the project, we knew that those promoters are regulated by FLGC and housekeeping RNAPs. And we can measure this transcription in the lab. We can mix this FLG-AM intergenic region DNA sequence together with nucleotides, housekeeping RNAP, and increasing concentrations of FLGC. So we got the transcript and we run them on the gel, and as you can see, FLGC switches on both housekeeping promoters. But apparently, this intergenic region is more complicated than we initially thought. We noticed 2 sequences going in both directions that look like they're supposed to be recognized by flagellar RNAP. So we did the same transcription experiment, this time with flagellar RNAP, and we got transcripts from those putative flagellar promoters. And in this case, FLGC addition led to their repression. And now I'm going to talk about the potential role of antibiotic resistance in flagellar biosynthesis. I'm going to introduce MRA. MRA is a transcriptional activator and global regulator of intrinsic antibiotic resistance. It activates many genes responsible for intrinsic antibiotic response in bacteria when antibiotics or other toxic chemicals are present in the cell. But we also think that MRA might be important for flagellar regulation as well, because we found that MRA binds in this FLGB intergenic region. And in this case, MRA can control up to 14 genes in these operons, which comprise over a quarter of all genes required for flagellar synthesis. So this is a sequence of newly discovered flagellar promoters. You can see it's -10 elements on FLGA and FLGB. They also share -35 elements, which is quite rare. What was very unusual is the MRA binding site. And to explain you why this is unusual, let me remind you about two main classes of transcriptional activators. So activators usually act by contacting both RNAP and promoter DNA. And these connections are very important for the activators to do their job. So for example, in class 1 activators, they bind upstream of the -35 element and they contact alpha cell unit. In class 2 activation, activator binding site overlaps -35 element and it contacts sigma cell unit of the RNAP. And you can notice that in both cases, activator binds upstream of the promoter. So with this information, if we come back to the sequence, we can predict that MRA will be an activator of FLGA operon because it overlaps -35 element as canonical class 2 activator. However, this is not the case. MRA in our experiment, it activated specifically FLGB promoter. And in FLGB case, MRA binds between its -10 and -35 element. So here we thought that MRA have a distinct mechanism of transcription activation. So we're curious about this unusual mechanism. And as you remember, I told you at the beginning of my talk that sigma factor needs to separate two strands of the DNA to make an open complex. And we found out that MRA activates transcription by helping in smelting. So to visualize FLGB open complex, we did thermodynamic footprint assay. It's a method in which we can visualize distortions on DNA as a band. So in the second lane, you can see control, but as soon as we add MRA, it bends DNA and two new distortions appear, 5 base pairs on either side of MRA at positions -11 and -34. And this bottom base that gets distorted by MRA is actually quite important adenine in FLGB promoter. I will tell you why in a moment. But what you're going to see next is that if we add RNAP, flagellar RNAP by itself, it's very bad in unwinding FLGB. It unwinds FLGA much better. We can see that FLGB open complex is being formed, but it cannot be fully open. You can see that only position -9 and -8 are being slightly open. However, in all of the bacterial promoters, -11 adenine base unwinds first. This is exactly where nucleation of the open complex happens, which is a crucial step in transcription initiation that is not happening in the FLGB case. You can see that this -11 adenine is still closed if we add RNAP, which is by itself. However, if we add both MRA and flagellar RNAP together, now we get a proper unwinding of the FLGB. from the upstream edge at -11 to position +5 downstream of CSS. And this happens because RNA restores this canonical -11 adenine in FLGB promoter, which allows flagellar RNA to capture DNA strand for the stable open complex. And this is our model of FLGB open complex and interaction between RNA in yellow and RNAP in transparent. Only sigma factor is colored in purple. So according to our model, RNA binds approximately 90 degrees away from the open complex. It do not interact with any RNA subunit, but it bends DNA into conformation that is more preferable for flagellar RNA binding and stabilizing the open complex. So, in summary, we found that RNA binds within the FLGB intergenic region and it might control a substantial subset of flagellar genes. Unexpectedly, RNA activates contribution from the FLGB promoter via DNA distortion, which facilitates capture of the -11 adenine base, enabling stable open complex formation by flagellar RNA. Big thanks to Granger Lab and Browning Lab. Also, big thanks to Gavin Trust for funding my PhD studies and Microbiology studies for the travel grant. You can scan the QR code and read our paper. Thank you very much for listening, and if you have any questions.
That was great, thank you. So, do you have any questions?